All right, before we go into the cell cycle, let's first talk about deoxyribonucleic acid. Once again, deoxyribonucleic acid is just a long name for DNA. And it's a biomolecule, right? We talked about what are the four types of biomolecules. We have carbohydrates as one, proteins as another, lipids as another, and then finally nucleic acids as the final one. And DNA, it's found once again within the nucleus, and for some reason it's not going. Whoa, way too fast now. Okay, it's found within the nucleus. It's our genetic material. The decondensed form of DNA is called chromatin. So some parts of the chromatin are going to be more condensed than others, but still it's not as condensed compared to when it's a chromosome. Okay, so when we're talking about these chromosomes, you could see here how that's different. And you notice how here's the nucleus here. The DNA is kind of just around here. You can't really see the individual strands of DNA. While over here, you started to see that the DNA is a little bit darker and stuff. So it's more condensed in this case scenario. So when the chromosomes, especially when we're talking about like mitosis or meiosis, they're so condensed, and at this point in time, they are replicated. And when they are replicated, normally we'll have 46 chromosomes or 23 pairs, right? If you do 23 times 2, it's 46, right? So we have 46 chromosomes in the normal human cell, somatic cells. However, in our sex cells, our gametes, there's only 23 chromosomes in meiosis. We'll talk about that in just a second and later in the semester as well. But with this chromosome here, if you look at the structure on the picture right here, so here's a chromosome that's been replicated already. This is one DNA, one strand of DNA. Whoa, what in the world is going on? I'm not drawing that, by the way. This is one strand of DNA over here, and here's another strand of DNA over here. It's lagging. Okay, so these two strands are identical strands of DNA here, and they are linked together here at the centromere here. Okay, here's the centromere, our proteins that link the two sister chromatids. So this DNA here is called a sister chromatid, and this DNA over here is called another sister chromatid. So there's two sister chromatids for each chromosome. Alright, once again, it's linked together by these proteins called a centromere, joined together. Okay, so cell division is when the cell divides, like the name implies. For mitosis, this is where the somatic or body cell, this is for most of our cells in our body, is called somatic cells. They will end up making two identical daughter cells. So like this. This is one body cell, ends up making two identical daughter cells at the end. Okay, so once again, how many chromosomes is that? It's going to be 46 chromosomes in each cell. This is different from, say, meiosis. Meiosis is when you have one cell gets divided once, and then it gets divided a second time to make a total of 4 cells. Okay, so it ends up making our gametes, and there's 4 cells, and they're not identical because these cells only contain 23 chromosomes each. Okay. Gametes are our sex cells, also known as the sperm for males and for females, eggs. Okay. The cell cycle, if we're talking about mitosis, is going to consist of interphase and mitosis. You'll notice from this picture over here, look how small mitosis is in compared to interphase. So most of the cell cycle is going to be composed of interphase, and just a small part of it is going to undergo mitosis. Not all cells undergo mitosis too. Most cells will undergo mitosis to replace damaged cells or to end up increasing the number of cells, like when we're growing. But some cells, they never undergo mitosis after they maintain its shape, like the neurons. Most neurons don't undergo mitosis. Interphase, once again, is most of the cell cycle, and it's where it's normally going to be doing its normal daily metabolic activities and so on and so forth. There's different stages within the interphase. There's G1 phase. This is where the cells continues to grow and produce new organelles. It ends up starting to prepare DNA replication by replicating the centrioles. Remember, the centrioles are the ones that will be part of the centrosomes to make the mitotic spindles. And it will end up stopping at this location if the DNA is damaged. You don't want to continue further and replicating DNA if it's damaged, right? You don't want cancer to occur. So it stops it until it repairs that DNA. Some of the cells ends up going to this alternative stage where they don't undergo undergo mitosis anymore. So once again, most neurons don't undergo mitosis. S phase is where they end up replicating the DNA to make the two sister chromatids. And then the G2 phase is continuing to repair any kind of DNA errors from the DNA replication and helps prepare the cell for cellular division. The centrioles at this point in time will finish replicating so that they could end up making those mitotic spindles later. You'll notice that this cell here is and this cell here are in interphase because you can still see the nucleus here and you don't see the individual chromosomes yet. Okay, this is the stages of mitosis. Once again, very short in comparison to interphase for the cell cycle. And once again, the other thing that's important is interphase part of mitosis? No. Interphase is part of the cell cycle, but it's not part of mitosis. The very first stage of mitosis, prophase, what happens there is that the chromatin starts to condense into chromosomes. Remember, the chromosomes has those two sister chromatids joined together by that centromere. So if we're going to draw it again, here's the sister chromatid, here's the centromere, here's another sister chromatid over here. And besides that, the nuclear envelope will start to disappear along with the nucleolus. The centriole starts to move towards the pole, as we can see over here. The centriole or centrosomes are going to be moving towards the pole, and mitotic spindles are going to be starting to extend from it. Okay. So over here, notice how the chromosomes, you can start to see condensing. Don't really see a nuclear envelope anymore at this point in time. Metaphase is where the chromosomes ends up aligning itself at the metaphase plate or equatorial plate. The spindle fibers will end up starting to attach to the centromeres. Okay, specifically, it doesn't attach to the centromeres because there's these proteins next to the centromeres called kinetochores. Don't worry about that term. But just think of it as attaching at the centromeres. Technically, it's not, but just think of it as it is. After it's attached to those kinetochores on the centromeres, as you can see over here, it's connected over here. That's metaphase, and then it's going to start pulling it away and breaking the centromere, and that's anaphase. So the spindle fibers that are attached to the centromere starts to pull. It'll split the centromeres. So instead of sister chromatids, they are now called daughter chromosomes. So each one of these are called daughter chromosomes here. And they go to the opposite poles. At this point in time, cytokinesis is starting to begin. And what cytokinesis is this pinching of the cell here, okay? And this pinching of the cell will cause this structure to call a cleavage furrow to form. You notice that the cleavage furrow is clearly seen over here in telophase. Telophase is when the nuclear membrane starts to form. It's basically the opposite of prophase. So prophase, the nuclear membrane disappeared, right? In this case, the nuclear membrane forms. The nucleolus starts to form. And also the chromosome starts to decondense back into chromatin. The mitotic spindles will start to dissipate and cytokinesis will eventually complete. And when cytokinesis completes, you have two identical cells. Okay, that's it for this chapter, but before you leave, let me just show you an animated, crude animation of mitosis real quick. I'm going to go all the way down. Okay, just repeat what are the stages, what's going to happen um, throughout the stages. Okay, well, first off, what stage of the cell cycle are we in at this point in time? This is interphase, right? We still see that the nuclear envelope is still there, the nucleolus is still there, and also the DNA that you see over here, it's still chromatin, right? It's not condensed, it's in its loose form right now. Okay, so this is interphase. Okay, so what happens in prophase? You should know. Okay, so what's the next step? When during mitosis, prophase, what's going to happen? The nuclear envelope is going to disappear. The nucleolus is going to disappear. The chromatin will start to condense into chromosomes, right? Once again, chromosome has those two sister chromatids joined together by the centromeres. And what's going to happen to the centrioles here, or the centrosomes? They're going to move to opposing sides of the cell. So here, it's going to animate this. So chromatin becomes chromosomes. Nucleolus disappears, nuclear envelope disappears, and we have the centrosomes going. Oh, okay, it starts to make spindle fibers and also starts to move to opposing poles. That's prophase. Okay, so after prophase, we have metaphase. What's going to happen to metaphase? The chromosomes will end up aligning at the equatorial plate or metaphase plate, as well as the spindle fibers are still starting to join at the kinetic cores of the centromeres, right? So those are the mitotic spindles going to the kinetic cores of the centromeres. That's metaphase. Anaphase, what's going to happen? Those mitotic spindles will start to pull and then break the centromeres so that the sister chromatids will become daughter chromosomes. So over here, breaks apart daughter chromosomes, and then it's going to pull the daughter chromosomes to the opposing poles. At the same time, what's going to happen in anaphase, the beginning of this process called cytokinesis, right? So cytokinesis is going to occur, and then this last stage is going to be telophase. Telophase is going to do the exact opposite of prophase, so the chromosomes are going to be decondensed into chromatin. The nuclear membrane is going to reappear, nucleolus reappear, mitotic spindles disappear. Okay, so all that's going to occur. So nuclear envelope appears, chromosomes decondense into chromatin, nucleolus reappears, decondense of the mitotic spindles, and you're going to finish off what process? Cytokinesis. Now we have two identical daughter cells. Just to reiterate some stuff, once again, the cytokinesis portion of it, what kind of cytoskeletal subunits causes this to occur, to cause this pinching of the cleavage furrow here? And if you say microfilaments, you are correct. What about the mitotic spindles that we just to pull the chromosomes away from each other? What kind of cytoskeletal subunits was that? Those are the microtubules once again, and so on and so forth. Okay, good. Okay, that's it for this chapter.